Dear students, today we will learn about photosynthesis. The photosynthesis will be a three-part series lecture. The first part will begin today. Enjoy this first part, and I am your Rajkumar sir. Topics that will be covered in the first part is the introductory part of introduction about the photosynthesis, chloroplast, photosynthetic pigments, nature of light, and the historical update in brief. Now, what is a photosynthesis? The overall process by which plants, algae, and prokaryotes use light energy to synthesize organic compounds is called photosynthesis. It's an anabolic process where small molecules are combined together to produce complex molecules. Again, it's an androgonic process, that is, it stores energy. It uses carbon dioxide, water, minerals, chlorophyll, light energy in the form of photons to produce carbohydrates, that is food, which is in the form of glucose, and oxygen, which is a byproduct of the photosynthesis and which is used for breathing by one and all. This is the summarized form of the photosynthetic reaction. CO2 plus H2O in the presence of sunlight and chlorophyll gives you C6H12O6 plus. The photosynthesis consists of two types of reactions. One is light reactions that occur in thylakoids, which converts light energy to NADPH and ATP. The second part is the dark reactions or the Calvin cycle that occurs in stroma, and it uses NADPH and ATP to fix carbon dioxide to make sugars. And the two types of reactions occurs in the chloroplast. However, they are segregated in space. The light reactions, they occur in the thylakoids, and the dark reactions, they occur in the stromal or the matrix of the. Now, what is the importance of photosynthesis? Photosynthesis is all bottled sunshine, meaning that the life which is occurring on the earth is directly or indirectly dependent or constructed from the sun's energy. The use of energy from the sunlight by photosynthesis is the very basis of life on the earth. The product of the photosynthesis, that is the food, it is the food for all autotrophs and all other heterotrophs. It produces biomass. The fossil fuels, they are the product of photosynthesis. All the oxygen that is produced on the earth is the result of the photosynthesis. Research for photosynthesis in vitro conditions is going on, so that one day we will be able to replicate the process and in its entirety in vitro conditions, and we will be able to synthesize food in vitro conditions. The photosynthetic research is also important because of the mechanism of action of herbicides, because they block the light reactions. Some of the herbicides, we will be dealing with these herbicides in the second part. The carotenoids, they protect chloroplast from the photooxidative damage. Now coming to the photosynthetic yield. If the year's yield of photosynthesis were amassed in the form of sugar cane, it would form a heap over 2 miles high and with a base of 43 square miles GE4. And if all of this sugar cane were converted into sugar cubes, 0.5 inches on one side and stacked end to end. The sugar cubes would extend 1.6 into 10 raised to power 10 miles or to the planet Pluto. See the amount of photosynthesis. The photosynthesis by green plants on the land is 50 into 10 raised to power 9 tons per year, and by ocean plants it is 180 into 10 raised to the power 9 tons per year. The plants that are growing in oceans and other freshwater algae accounts for the 90% of the total photosynthesis. You might be thinking that the photosynthesis on the land is important. No doubt it is important, but it contributes only 10% of the photosynthesis. The major part is done in the oceans and other freshwater ecosystem. Now coming to the structure of the leaf, you are aware of the structure. This is a typical dicot leaf structure, the 3D model, 3D image. In this you see an upper epidermis which is bounded by a cuticle and a lower epidermis having stomata in them with a lower cuticle, and in between these two layers you will see the mesophyll cells. Mesophyll cells are of two types. The upper one, which are vertically elongated, are called as the palisade mesophyll cells, and the lower one, it is called as the spongy mesophyll cells. The palisade mesophyll and cells and the spongy mesophyll cells, they contain chloroplast, because of which they are green in color. The chloroplast contains chlorophyll. As a result, the leaves, they are green in color. Now coming to the chloroplast. Chloroplasts are the center for photosynthesis. They are the site for photosynthesis. And if you see a transmission electron micrograph of the chloroplast, you will see that it is bounded by an outer membrane and has an inner membrane. There are two membranes of the chloroplast, and that forms the chloroplast envelope. In the center of this, bounded by the chloroplast envelope, is the stroma that contains 70 S ribosomes and naked DNA. Interspersed between the stroma are present the grana and the thylakoid membranes. They are the sac-like structures. Some of the sac-like structures, they are stacked one over the other, like the piles of the coins stacked one over the other, and forms the grana. The stroma also contains, besides the 70S ribosomes, a naked circular DNA, the starch grains, the oil droplets, which are the results of photosynthesis. Now you should see the structure, how the structure of the chloroplast assists its function, differences between the chloroplast and the mitochondria. This is a detailed structure of the chloroplast, where you can see the grana thylakoids and the stroma thylakoids, or the stroma lamellae. The grana, they are the stacks of thylakoid, just like coins placed one over the others. In fact, these are not solid coins, they are hollow coins. With Now coming to the photosynthetic pigments. Photosynthetic pigments are located in the chloroplast thylakoidal membranes. अगर आपसे पूछा जाए कि फोटोसिंथेटिक पिगमेंट्स जो क्लोरोफिल्स, कैरोटीनॉइड्स और फाइकोबिलेंस होते हैं, कहाँ प्रेजेंट मिलेंगे, कहाँ मिलेंगे, where you will find the photosynthetic pigments, you will find them in the thylakoid membranes. Thylakoid membranes कौन सी थी? दो तरह की थी. One was the grana and the second one was the stroma thylakoids. नाउ कमिंग टू द क्लोरोफिल्स. क्लोरोफिल्स देखें तो क्लोरोफिल्स जो हैं, ABCDE। और इसके अलावा देखें तो बैक्टीरियो क्लोरोफिल होता है, जिसको कि आप अलग बैक्टीरियो क्लोरोफिल ABCD और बैक्टीरियो विरिडिन में क्लासिफाई कर सकते हैं। They are found in all photosynthetic organisms, but but all the organisms they contain a mixture of more than one kind of pigments. Chlorophyll A and B they are abundant in green algae and higher plants. There, there are several spectrally distinct forms of chlorophyll A, and these are shown represented by their absorption peaks, like chlorophyll A 670 to 673, chlorophyll A 680 to 683. Besides this, there are special types of chlorophylls which are designated as P700 and P680. And P700 and P680 they serves at the reaction centers of photosystem 1 and photosystem 2 respectively. Now coming to the chlorophyll molecules, as I told you that they are located inside the thylakoid membranes, and chlorophylls they have magnesium in the center. Chlorophyll pigments harvest energy, photons, by absorbing certain wavelengths, blue and red, which are absorbed the most. No doubt the chlorophylls they absorb all the wavelengths, but the maximum light that is absorbed is present in the blue and red regions. That is, blue wavelength is 420 nanometers, red wavelength is 660 nanometers. That is why you will observe a peak, the absorption peaks at these wavelengths. The plants are green because the green wavelength is reflected and it is not absorbed. The rest of the wavelengths they are absorbed and the green wavelength is reflected. So the plants they appear green. All chlorophyll consists of a tetrapyrrole ring having conjugated double bonds and a phytol chain. Phytol chain, it's a long chain alcohol. The center of the tetrapyrrole ring has a magnesium coordinatively held to the nitrogen atoms of pyrrole rings. Side chains they are attached to pyrrole ring vary in different chlorophylls. Phytol chain imparts the chlorophyll molecule the lipid solubility and water insolubility. Because of this phytol chain, the chlorophyll molecule is lipid soluble and it's water insoluble. Chlorophyll B is a minor chlorophyll in higher plants and green algae. Its absorption peaks are 455 and 640 nanometers. See, in this picture also, you see that there is a difference in chlorophyll A and chlorophyll B molecules. In case of chlorophyll B, the methyl group is replaced by an aldehydic group. This is the porphyrin ring. You can see that in the tetrapyrrole ring there are conjugated double bonds which are very important in absorbing the energy. Methyl, chlorophyll A has a methyl group, chlorophyll B has a carbonyl group. And on the right hand side you can see the atomic model. Now coming to the second type of pigments, the carotenoids. Carotenoids are also important pigments responsible for photosynthesis. They are the supplementary light receptors. They absorb light energy and they pass on the light energy to the chlorophyll molecules. They are yellow, red or purple pigments. Beta carotene is the most important one. It's a red-orange isoprenoid compound. It's a precursor of vitamin A in animals. Xanthophyll is yellow. Absorption peaks ranges from 400 to 500 nanometers. It protects the chloroplast against photooxidative damage. Xanthophylls like lutein and zeaxanthin are oxygen derivatives of carotenes containing 1 to 8 oxygen atoms. Now coming to the next one, the phycobilins or the phycobilly proteins. Phycobilly proteins are the water-soluble proteins present in cyanobacteria and certain algae like rhodophytes, cryptomonads, glaucocystophytes. They capture light energy which is then passed on to the chlorophyll during photosynthesis. Phycobilly proteins, they are formed of a complex between proteins and covalently bound phycobilins that act as chromophores, the light capturing part. Their most important constituents are the phycobilisomes, linear tetrapyrroles. They have a linear tetrapyrrole structure that have extended polyene system found in chlorophylls but not their cyclic structure or central magnesium. Phycoerythrin and phycocyanin. They have absorption peaks respectively in 409 phycoerythrin having an absorption peak of 490, 546, 570 nanometers. Phycocyanin having an absorption peak of 608 nanometers. You see that these pigments they absorb all sorts of pigments. No doubt, if you see the absorption pattern, you will see that it absorbs a wavelength of 500 nanometers, say 540 nanometers, 530, 510 nanometers, but the maximum absorption is in this region. The absorption also depends upon the solvent also. If you vary the solvent, the absorption peaks shifts on the right hand side or the left hand side. It all depends upon the solvent also. Phycoerythrin is a red protein pigment complex from the light harvesting phycobilly protein family present in red algae and cryptophytes. It is an accessory to the main chlorophyll pigments responsible for photosynthesis. Red pigment is due to the prostatic group phycoerythrobelin, which gives phycoerythrin its red color. Phycoerythrobilin is a typical chromophore in phycoerythrin and it is similar to the porphyrin of chlorophyll, for example, but the tetrapyrrole is linear, not closed into ring with a metal ion in the middle. On the right hand side, you see the red algae gracilaria, where from this pigment is iso. This is the structure of phycocyanobelin. Now coming to xanthophylls, as both are carotenoids, xanthophylls and carotenes, they are similar in structure, but xanthophylls contain oxygen atoms, while carotenes are purely hydrocarbons which do not contain oxygen. Their content of oxygen causes xanthophylls to be more polar in molecular structure than carotenes and causes their separation from carotenes in many types of chromatograph. Carotenes are usually more orange in color than xanthophylls. Xanthophylls present their oxygen either as hydroxyl groups and or as hydrogen atoms substituted by oxygen atoms when acting as a bridge to form epoxides. This is the structure of cryptoxanthin and this is the structure of a xanthophyle called lutein. Why plants are green? This is shown in this picture also because most of the light it is absorbed but it reflects the green wavelength or the green light. Now coming to the wavelength of light or the visible portion, it ranges from 400 to 700 nanometers. Short waves having more energy and long waves having less energy. Now the absorption of light by chlorophyll. Chlorophyll absorbs blue, violet and the red light the best. You can see the absorption peaks. They appear in the blue region and the red region. This is the absorption of the light by chlorophyll. This is called as the absorption spectrum. Now the question is, during the autumn season or the fall, what causes the leaves to change colors? You see all sorts of colors during the ऑटम सीजन, जब पतझड़ आता है तब आपको पत्तों के अंदर अलग अलग तरह के रंग दिखते हैं। उसके पीछे वजह क्या है कि क्लोरोफिल के साथ साथ दूसरे पिगमेंट्स भी प्रेजेंट होते हैं पतझड़ के दौरान। ड्यूरिंग फॉल और ड्यूरिंग ऑटम सीजन green chlorophyll pigments they are greatly reduced and they reveal the because of which the other pigments they appear, because the green chlorophyll pigments they mask the in the normal course they mask the effect of the other pigments. Carotenoids and the are the pigments that are either red, orange or yellow. They appear when the chlorophyll amounts decreases. Now coming to the nature of light. Light shows both particulate and wave nature. A simple equation relates the wavelength, frequency nu and speed or velocity of the wave. C is nu lambda. Light is a transverse electromagnetic wave in which both electric and magnetic fields oscillate perpendicularly to the direction of the propagation of the wave and at 90 degree with respect to each other. Light is also a particle which is called photon, having an amount of energy called quantum, plural is quanta. The energy of the photon is shown by Planck's law. E is h nu, E is equal to h nu, where h is the Planck's constant 6.626 multiplied by 10 raised to the power minus 34 joules per second, where nu is the frequency of light. Sunlight is just like a rain of photons of different frequencies. Our eyes, they are sensitive to a very small range of light, that is the visible light, which ranges from 400 nanometers to 700 nanometers. Wavelength and the frequency, they are inversely proportional, as we had seen in the previous slide. This is the entire electromagnetic spectrum, and different wavelengths of the visible light they are seen by human eyes as different colors. जो अलग अलग वेवलेंथ होती है जैसे 400 नैनोमीटर्स की, 500 नैनोमीटर, 600 नैनोमीटर, 700 नैनोमीटर्स, वो हमें कैसे दिखती है, कैसे महसूस करते हैं हम उनको अलग अलग कलर्स से महसूस करते हैं। इनफैक्ट जो है कलर्स प्रेजेंट नहीं है नेचर में, ये कलर्स नहीं है, हमें कलर्स अपीयर होते हैं बिकॉज ऑफ आवर ब्यूटीफुल आईज। ये पूरा स्पेक्ट्रम देखेंगे जहाँ गामा रेज है, एक्स रेज हैं, यूवी रेज हैं, इंफ्रारेड रेज हैं, माइक्रोवेव्स हैं, रेडियो वेव्स भी हैं, उसमें सिर्फ छोटा सा पार्ट, बेहद छोटा सा पार्ट जो है विजिबल स्पेक्ट्रम का आता है, जो कि 400 नैनोमीटर से 700 नैनोमीटर तक होता है। ये इलेक्ट्रोमैग्नेटिक स्पेक्ट्रम और विजिबल लाइट्स हैं जहां पर अलग अलग पर्पस के लिए इनको यूज करते हुए भी आपको दिख रहा है। जहाँ गामा रेज हैं बड़ी डेंजरस हैं, एक्स रेज हैं, यूवी रेज हैं, इंफ्रारेड माइक्रोवेव्स और रेडियो वेव्स, आपको इनके पर्पस भी दिख रहे हैं। नाउ कमिंग टू द अब्जॉर्प्शन स्पेक्ट्रम। अब्जॉर्प्शन स्पेक्ट्रम क्या होता है? It relates the amount of light energy taken up or absorbed by a molecule or a substance with respect to the wavelength of the light. The absorption spectrum of chlorophyll A and chlorophyll B shows that these pigments chiefly absorb blue and red regions. Chlorophyll absorbs blue, violet and the red light the best. The exact position of peaks depend upon the solvent which is used for extraction. The transmitted and the reflected lights, they are vastly rich in green, for which is in 500 to 600 nanometers, giving the leaves their green color. Now this is the action spectrum. It shows the magnitude of photosynthesis टू लाइट, यानी कि फोटोसिंथेसिस जो है एग्जैक्टली होते होते रेट ऑफ फोटोसिंथेसिस इज प्लॉटेड अगेंस्ट द different wavelengths, then we get the action spectrum. Now the question is how you measure the photosynthesis. The magnitude of photosynthesis is measured and plotted by the amount of action. What is the amount of action? It's the rate of carbon dioxide fixation, rate of oxygen production, NADP reduction, changes in the biomass. Now, if, if we add the absorption spectrum of the chlorophyll, you can see that they almost coincide with each other. They correlate with each other, because of which we can easily say that the rate of photosynthesis is maximum in the 400 and in the blue and red regions. Now coming to the historical update. The history start. This will be in a brief. If you want to study it in detail, you can go consult your books also. Aristotle and Theophrastus in 320 BC plants said that plants derive nutrition directly from the soil. Van Helmont in 1648 showed that all food comes from the green plants and it comes from water also. Stephen Hales in 1723, green plants require sunlight for their nutrition. Joseph Priestley in 1772 said that the green plants purify foul air, which is called as dephlogiston, produced by the burning of the candle, and it converts it into pure air by producing phlogiston, that is oxygen. Ingenhouse in 1779 found that the plants purify the noxious air only in the presence of light. Lavoisier identified pure air produced by plants as oxygen and foul air as carbon dioxide in 1783. Nicolas de Saussure in 1804 found that water was used in photosynthesis. Dutrochet in 1837 found that chlorophyll is important for photosynthesis. Liebig in 1845 showed that the main source of carbon in plants is carbon dioxide. Van Meer experiment showed that the green plants transform light energy into chemical energy of organic matter in 1845. J.V. Sachs in 1862 observed that the first visible product is starch and chlorophyll is present in chloroplast. Engelmann in 1888 found that the chloroplast is the seat of photosynthesis and plotted the first action spectrum using spirogyra. This is the action spectrum for photosynthesis. What he did, that in place of, he chose bacteria that were attracted by oxygen and placed the spirogyra on the slide, and that slide was put up on the stage of the microscope, and in place of illumination he substituted it by a microspectral apparatus, which, as the name implies, produced a tiny spectrum of colors that it projected upon the slide under the microscope, and then he arranged a filament of an algal cell parallel to the spread of the spectrum. The oxygen-seeking bacterium congregated mostly in the areas where the violet and the red wavelengths fell upon the algal filament. Blackman in 1905 found by his experiments the existence of light and dark reactions, or two stages of the photosynthesis. He proposed the law of limiting factors. Van Niel in 1931 found that some bacteria uses H2S instead of water in photosynthesis. Photosynthetic reactions are redox reactions. Carbon dioxide plus H2S plus light energy gives you C6H12O6 and water and plus sulfur granules. Emerson and Arnold in 1932 by flashing light experiments at different temperatures found the existence of light and dark reactions as predicted by Blackman. Robert Hill in 1937 showed that the chloroplast causes photooxidation of water to produce a hydrogen donor. Rubin, Hassett and Kamen in 1941 using labeled oxygen conclusively provided evidences for the oxygen evolved comes from the water and not from carbon dioxide. Arnon, Allais and Watley in 1954 used labeled carbon dioxide to show that the carbon dioxide is fixed by isolated chloroplast. Emerson et al. In 1957 found the Emerson enhancement effect and the red drop. Melvin Calvin found the Calvin cycle or the C3 cycle or the dark reactions and was awarded Nobel Prize for chemistry in 1961. Hill and Bendel proposed the Z scheme and suggested that the two photosystems operate in series. Arnon 1961 found the photophosphorylation. Peter Mitchell discovered chemiosmotic coupling hypothesis in 1961 was given the Nobel Prize in Chemistry in 1978. Hatch and Slack cycle was or the C4 cycle was discovered in sugarcane in 1966. Disenhofer, Huber, and Hartmut-Marshall West Germany crystallized the photosynthetic reaction centers of bacterium Rhizobacter viridis. Rudolf A. Marcus, 1985, Theory of Electron Transfer Reactions in Chemical Systems. Nobel Prize in Chemistry in 1992 for his contributions to this field. The electron transfer reactions are rapid as fast as few picoseconds and they are specific. Now revise this topic and enjoy learning. Thank you. You can uh, email your inquiries to me.